Hello everybody, welcome to YouTube channel Enriched Biology. Today we'll do the anatomy of the leaf. Previously we did the anatomy of dicot root as well as dicot stem. And anatomy of leaf. You know leaves they are of two kinds. Uh, Dicot leaves and monocot leaves. The dicot leaves they are dorsiventral and monocot leaves they are isobilateral. Dorsiventral means there are two surfaces they are distinguished. One is bright and glabrous and another is somewhat rough, uh, dull. Another is somewhat dull. So they are the dorsiventral leaf. So we will do the anatomy of dorsiventral leaf, that is dicot leaf. If you cut the transverse section of dicot leaf, dorsiventral leaf, that is of sunflower or of mustard, you will under the microscope you will see the following layers. There are two epidermis, upper epidermis and the lower epidermis. Upper epidermis, it is on the adaxial surface, lower epidermis on the abaxial surface. So epidermal cells, they are compactly arranged without intercellular spaces. However, they have the little pores which are called as the stomata in both epidermis, upper epidermis as well as the lower epidermis. These stomata, they are more on the lower surface, that on the abaxial epidermis than on the adaxial epidermis. That is, sometimes in adaxial epidermis, the stomata they are lacking as well. So what are these stomata? These stomata, they are the pores. They are connected with the tissue of the cell, that is the cortex tissue of the cells. They help in exchange of gases as well as they also, perform the function of transpiration. So when they are opened, then there is transpiration. When they become there is transpiration, and when they are closed, there is no transpiration. So the stomata, they are surrounded by two bean-shaped cells, kidney-shaped cells. These are the cells. These are called as the guard cells. Stomata, they are surrounded by two guard cells which are bean-shaped or kidney-shaped. That when they become turgid, the stomata becomes open. When the guard cells they lose water, and the stomata will become closed. They will they will be non-turgid. There will be no water here, and then the stomata become closed. These guard cells they are surrounded by some other cells which are called the subsidiary cells. So water is translocated from the subsidiary cells to the guard cells. So then, in addition to this, epidermis is also covered by a layer of cuticle to prevent more desiccation and to protect the plant. Then this both epidermis, upper epidermis as well as the lower epidermis, they enclose the tissue, parenchymatous tissue, which is called as the mesophyll. This whole parenchymatous tissue is called as the mesophyll. Mesophyll is distinguished into two types. Upper layer of the mesophyll, it consists of elongated parenchymatous cells which are loosely arranged below the upper epidermis in 2 to 3 layers. This form the palisade tissue, or you can say palisade parenchyma. The lower layer which are present near the lower epidermis on the abaxial surface, the cells they are loosely arranged and large air cavities are present in between the cells. So they form the spongy structure, and this layer is called as spongy parenchyma. This is called as the spongy parenchyma. So in this case, upper epidermis, lower epidermis, and mesophyll is present, distinguished into palisade and the spongy parenchyma. Vascular bundles, they are arranged along the midrib as well as along the veins. On the veins, the vascular bundle, they are of the equal size and strength, but on the midrib, they are somewhat larger. So each vascular bundles, they are, as in case of stem, they are endark. Protoxylem is facing towards the center, towards the pith, and metaxylem towards the up to say pericycle toward the cortex. Then these bundle sheath, vascular bundle, they are conjoint. Xylem and the phloem, they are present on the same radius. They are present on the same level. But they are not open. No arm to say cambium is present. Vascular bundles, they are surrounded by Schleeren chymatous bundle sheath. They are surrounded by Schleeren chymatous bundle sheath. This is all about the histology or anatomy of the dicot leaf or dorsiventral leaf. The anatomy of that, to say, monocot leaf is similar to the dicot leaf with some differences. In this case, what happens? Again, epidermis, upper epidermis and lower epidermis are present. They enclose the mesophyll tissue, but mesophyll tissue, it consists of only spongy parenchyma, no pellicate tissue are present. It consists of spongy parenchyma, and the vascular bundles, they are again, to say, surrounded by bundle sheath of the Schleeren chymatous cells. They are conjoint and dark. They are conjoint and dark. To say number of stomata are more in the axial surface than on the adaxial surface. They are present on both the sides. Then in some, what happens on the adaxial surface, some of the cells, they become elongated, transparent cells which are called as the bulliform cells. When these bulliform cells, they become turgid, they extend, the leaf is open. When the bulliform cells, they become flaccid due to loss of water, the leaf will curl down, it will be wilted. So this is all about the, to say, histology of monocot leaf. Monocot leaf is called as the isobilateral. It has no different, to say, abaxial surface and the addaxial surface. They are, both the surfaces, they are equal, and this is all about the histology of monocot leaf. So then we also did the secondary growth in case of dicot stem. You told in the dicot stem, the bundle are, vascular bundles are, they are collateral. They are conjoint. So the in between the xylem and the phloem, they are present at the same level, they are present at the same radius, and cambium is present in between the xylem and the phloem. That is, open vascular bundles are present. That cambium is primary cambium, that is also called the intravascular cambium. And secondary cambium arises between the two bundles, to say, and then a ring is formed. In case of root, you know, the vascular bundles, they are present at the different radii. In this case, the vascular bundles, they are radial. Phloem and the xylem, they are present at the different radii. At one radius, on the outer side, phloem are present. Then on the alternating with the phloem on the second radius, in the inner to the phloem, the xylem elements are present. So such type bundles, they are called as the radial. So what happens in this case, primary cambium is not present, as in case of dicot stem. So in dicot root, primary cambium is not present. Cell of the pericycle, some of the cells of the pericycle above the phloem and below the metaxylem. Below the phloem and above the metaxylem, they become, yes, active. They get dedifferentiated and produce the cambium. Some of the cells of the pericycle below the phloem and above the metaxylem, they become dedifferentiated and become active and regain the power of division, and they form the cambium. So a wavy cambial ring is formed. Like this, it is just like phloem, then it will, you can say, if it is rising below the phloem, then next it will rise above the metaxylem. Then below the then above the mesoderm. So in this way, wavy ring is formed. Wavy cambium ring, and this cambium is secondary cambium. It is entirely secondary cambium because it arises by dedifferentiation of the differentiated cell. This cambial ring become active, and after some time it becomes straightened and become the normal circular cambial ring. This ring adds xylem, secondary xylem toward the inner side and secondary phloem towards the outside. So xylem elements, they are added more than that of the secondary phloem. So this is all about the to say secondary growth in case of root. So in case of root, the cambium, vascular cambium is entirely secondary. In case of stem, the vascular cambium is partly primary and partly secondary. So this is all about secondary growth. And we have done the cork also. Cork is also formed by the cambial ring, which is cambium, which is formed by the dedifferentiation of the cortex cells. Some of these cells of the cortex, they become dedifferentiated. Why cork is formed? Because when more xylem is added in the root and xylem is added in the stem, in case of woods, trees, so it will exert pressure on the cortex. Ultimately on the epidermis, the cells will get obligated. So to protect the cell, then cork is formed. So what happens? The cells of the cortex, they become dedifferentiated, and these dedifferentiated cells, they are called as the cork cambium. They are also called as the phallogen. Phallogen, it divides and redivides. It adds cell towards the outside, which form the cork, that is, which is called the phallum. And the cells which are added toward the inside, they form the secondary cortex, which is called as the phalloderm. Phallum, cork cell, then cork cambium, phallogen, then the secondary cortex, phalloderm. Together they are called as the periderm. They are called as the periderm. So in this way, the cells are added towards outside, which are by the phallum. Though those get lignified, those get subrized and form a hard core above the plant. That bark you might have seen if you see the large trees, where outer portion is very much hard. And then you can say, what happens sometimes, some of the cells, which are the cork cells, they are parenchymatous and they are not lignified, and they get burst and form a pore in the cork. So say they are thin-walled cells, they are parenchymatous cells of the phallum. They are not lignified, they are not subrized, and they exert pressure. By the pressure of the inner cell, they get ruptured and form a pore. And the lens-like pore is formed, that is called as lenticle. These lenticles help in the respiration of the plant. So this is all about secondary growth, and this is all about the anatomy. In the next video, we'll do the important points to remember about plant morphology and plant anatomy. I hope you might have followed it, like it, describe it, and share with your friends. Okay, have a good day.